mention that both Brian and Lindsay have been named rising stars in proteomics and metabolomics by Journal of Proteome Research this year. So help me welcome Lindsay for the second part of the program.
I'm super excited to be here again, even though we can't be up in Boston. It's always a fun crowd, and following Brian up is definitely a challenge, but we're going to try to give you a little bit more in-depth discussion about some of the things that Brian talked about. And especially, I'm going to be trying to frame these within this software tool called Skyline. So we're going to have some hands-on with Skyline later. Try to download those files if you haven't already. And hopefully the TAs can help you get set up if you're having issues installing Skyline. I also have a couple disclosures. As Olga just said, I'm currently a postdoc at the University of Pennsylvania, but I also have co-founded and currently consulting at a company called Talis Bioscience. So as I had mentioned, I kind of have three main objectives for everyone today. So the first, I want to revisit some of those core concepts that Brian gave you a theoretical framework for this morning. And I want to reframe them using the Skyline ecosystem. So the two kind of main things here are going to be developing DIA assays and visualizing DIA data. I want to give us a little bit of a deeper understanding for quantitative liquid chromatography mass spectrometry data, including how chromatogram peaks are processed in Skyline. So Brian talked a little bit about how the data can be analyzed. We're going to try to give you a little bit more background on that so that you have a better intuition for what this black box is doing with your data. And then throughout, I'm going to have a lot of deeper dives, a lot of references for you to follow up on, including things like Brian talked a lot about this idea of index retention time standards, retention time alignment. I'm going to throw a whole bunch of references for you to dive a little deeper if that's something that you're interested in. So throughout the talk, the first part of the talk, I'm going to have anything that's relevant to Skyline as the software is going to have this kind of blue background with the white text. Just to orient you really quick, and if you'd like, you can follow along with Skyline open on a second monitor or this playing in the background while you look at Skyline. But kind of the basic structure of the Skyline program when you open it, on the left-hand side, there's a window called Targets. That's where your proteins, your peptides, and your fragments are going to be. Up in the top right corner, you see this kind of squiggle, swirly blue thing. That's supposed to be a peptide secondary structure, an alpha helix. If you drop that down, you may notice that there's two other settings for Skyline. There's also a small molecule setting, and there's a mixed setting for peptides and small molecules. And then down at the bottom, in the bottom right, there's a series of target summaries. So the number of proteins, peptides, precursors, and transitions that are in your document. So we're going to come back to Skyline throughout the lecture portion of this, and then we're going to be opening up Skyline and playing with it ourselves later. But what Skyline does is it's this ecosystem for various informatics used in quantitative mass spectrometry. So when we have a mass spectrometry question that we want to ask, a proteomics question, usually we start with some sort of hypothesis. In a targeted setting, your hypothesis might be a specific protein of interest or a specific pathway of proteins of interest. In DIA, as Brian just told us, what's really nice compared to a PRM, a targeted study, is that you don't necessarily need to know what question you're asking when you acquire your data. Skyline also assists in the assay development section. As Brian had mentioned, if you're doing targeted, like a PRM, there's a lot of upfront assay development that you have to do for those specific proteins of interest. For DIA, there's a little bit less assay development upfront, but there are certain things that we do have to decide, like Brian talked a lot about different isolation window schemes that are going to make sense depending on what kind of instrument you have and what kind of samples you're working with. The scope of the experiment might dictate the kind of isolation window scheme that you're optimizing. Then we talked a lot about spectral libraries. So that's another thing that we can do in Skyline. We can make the spectral libraries. We're going to be talking a little bit about that. And Skyline won't tell you how to do the acquisition on your instrument. You have to set that up, but you can actually export methods from Skyline. Methods directly from Skyline. It can analyze your data, process it. There's also several core analyses built into Skyline that we're going to talk a bit about, and even post-hoc additional analyses, post-processing, using various tools, including there's an external tool option for MS Stats, or you can export your data from Skyline, bring that into the MS Stats R package, and perform any of those R functions based on the input that you got from Skyline. So Brian gave us this good overview, right? When we're doing proteomics, we start with some sort of tissue or biofluid that we're interested in. We take the proteins out of that system. We perform the enzymatic digestion. We end up with peptides for mass spectrometry. They're separated by liquid chromatography. They go through analysis on the instrument. So precursors are selected in a targeted analysis. That's a very specific precursor. In DIA, we widen out so we get a whole range of precursors. Measure the fragments. We end up with mass spectra from the instrument, but we can stack those mass spectra up into chromatograms. So let's focus on this top part first, right? So there's a lot of different ways that we can get our protein. If we're going from tissue, we might have to perform processes like tissue homogenization. If we're in cell culture, we have to lyse our cells. We may decide to do further processing like subcellular fractionation. Now we have proteins. We may further fractionate our proteins, and Isabelle Bludau is going to give us a little bit of a vignette on some interesting protein extraction processes called size exclusion chromatography to get at protein complexes. As Brian mentioned, we can do PTM enrichments. We can do immunoprecipitations for specific proteins. For enzymatic digestion, we have a whole nother set of assay development questions. What kind of enzyme are we going to choose? Trypsin is very common. And as Brian mentioned, if we're doing DIA, most of these analysis tools assume you're using trypsin. But there's a lot of other options out there. Or you may be doing things like immunopeptidomics, which don't need to be enzymatically digested. And now we have peptides, but they're not quite ready for mass spectrometry yet. We might have to do more enrichment, another round of PTM enrichment. We might have to do another round of desalting. So this is just cleaning up the peptides. A lot of times these upstream processes either involve detergents or salt that need to be removed before we can analyze that mass spectrometry. At the end of the day, we end up with this set of peptides, right? And in a targeted environment, you know a lot of these details. You know a lot of these details about the mass spectrometry prep. One of the first things that we have to do in Skyline is record some of these method sample preparation details in there. So Skyline, really the core of Skyline is making sure you have these settings correct. So some of the major setting features here. So this is up in the top left-hand side of your Skyline document. There's a settings tab. And if you go to settings, peptide settings, you'll see a window, pop-up window looks like this. It has multiple tabs here, things like digestion, prediction, filter, library, modifications, then finally quantification. So the very first thing, no matter what you're doing in Skyline, whether it's DIA or a targeted analysis, you want to set up the digestion and the filter. So some of these things, for example, the enzyme, you know that from just whatever you did in your sample prep. In the filter, in targeted settings, typically just using default settings in Skyline is perfectly fine. There's also under settings, there's settings peptide settings, and there's also settings transition settings. So here again, there's going to be a filter tab that we need to set up for targeted experiments. When we're doing DIA experiments, there's a whole nother set of settings, and Brendan's going to be going through these in much more depth when we do a DIA data analysis with him tomorrow.
Lindsay, there's a question. Can you add multiple enzymes if you're using a combo of trypsin plus something else?
Yeah, so you cannot add multiple enzymes. So it's just a drop down. You can't like check multiple boxes for multiple enzymes. But what you can do is you can create your own enzyme and just give Skyline the rules for your kind of like custom enzyme. So you can kind of not hack Skyline, but a little bit. It's a hack to make it accept multiple enzymes. That was a good question. Yeah, so we're going to be looking at all of these settings ourselves in depth in just a bit, but The final thing that I wanted to mention in these settings is under the settings, transition settings, under the full scan tab, there's this MSMS filtering section kind of in the middle. If you set the acquisition method to DIA and you go to the isolation scheme, you have the option to add an isolation scheme in Skyline. And what that allows you to do is you can calculate your own isolation schemes using Skyline's isolation scheme calculator. So this is another option, in addition to using the encyclopedia software Brian mentioned, also has an isolation window scheme calculator in it. This is just another option that'll let you do that. So I just kind of plugged in some of the options here. For example, Brian talked about doing that 400 to 1000 MZ range. We can plug in a 24 MZ window width. He talked about optimizing the window placement for those forbidden zones and how that differs for different types of samples, but you can check that box and it'll optimize the window placement for a whole proteome, not a phosphoproteome, just a regular proteome digest. The deconvolution, you can select the stagger, you can select the margins, just like he talked about before. And what that'll do is it'll populate this scheme here, which you can then make graphs for, or you could copy that into your instrument itself. So that's just a little bit of background, putting some of the things that Brian said into context, but I wanted to dive a little deeper now into the analyzing of the data. So now let's assume you filled out this windowing scheme or you followed the recommendations Brian gave you, you went to your instrument, you acquired your data. Now let's talk about processing that data in Skyline. So what's really nice, Skyline is vendor neutral. So it's going to take your raw spectra and you can process it all directly within Skyline. Brian also mentioned there's options where you can combine other software and pull that into Skyline. We'll talk a little bit about that. But I wanted to give you a bit of background into what exactly Skyline is doing with your data. So there's several steps when Skyline's reading in your data, and we'll again, be talking about this more in depth tomorrow, but the first step, Skyline is extracting chromatograms. Then it resamples the chromatograms, looks for where peaks might be in your data, groups them together, and then performs the peptide identification. So a little bit more on those. So very first thing, as we just heard, mass spectrometry data is three-dimensional, right? So we have retention time dimension. This is the peptides coming off of the liquid chromatography column. Then we measure the mass to charge for all of those peptides as they come up off the column. And then finally, we have a measure of intensity. So when we look at that overall, we get this kind of three-dimensional representation of our mass spectrometry data. If we take one set retention time and draw the slice of mass to charge and intensity data for that one retention time, we get the mass spectra. Conversely, if we hold the mass to charge set at one value and then extract the intensity across the retention time, we get the chromatogram. So Skyline's not thinking about the mass spectra, it's thinking about the chromatograms. So everything that Skyline's doing is kind of in that chromatogram space. So we set a mass to charge at a given value. We extract the intensity for that mass to charge over all of the retention time that we measured. We have a chromatogram extracted, an extracted ion chromatogram, XIC. So for the chromatogram, you might notice that I've drawn these small dots spaced along this black line. That's actually where the mass spectrometer made measurements. So each one of these points is one MSMS spectra. And all of our mass spectrometry data, these are not spaced entirely perfectly apart. It's not a perfect sampling. So one of the first things that Skyline's going to do is look at all these scans made by the instrument and resample it. So this is because downstream processes in Skyline, the scans have to be on an even interval. So Skyline's going to look at the data, it calculates an interval width that best fits your data. Does this using something called a linear interpolation. but it's going to look at the data and say, if I sample at this interval, I have captured the shape of the chromatogram and I haven't distorted the data. If the intervals are too wide, the shape of the peak would become distorted. If it's too narrow, then the processing time will get jacked up. It'll take a long time to process the data. So Skyline's going to look, it's going to interpolate using that linear interpolation, a resampling across these peaks. Now it's going to look through the chromatogram and try to determine where the peaks are. So Skyline uses a peak detection algorithm that's called crawdad. It's based loosely off of another previous McCoss Lab grad student, Greg Finney. Crawdad looks at the data, it finds the minima and the maxima across the chromatogram. So I'm kind of using these red dots to symbolize it's looking for the minimum, it's looking for the maximum, and then finally it looks for the inflection point, kind of the middle of that peak. So that's where the second derivative is equal to 0. If you remember back to your calc or pre-calc days, it finds minimum, it finds maximum, it finds the inflection point in the curve using the second derivative equal to 0. These set of points, minimum, maximum, and the middle, defines the detected peak. So you can kind of see this in Skyline a little bit. In Skyline, in the chromatogram window, if you right click and you go to transform, you can choose the second derivative transformation in your data. So crawdad's placing those peak boundaries in Skyline. It's going to set them initially at those inflection points. So remember the inflection points where the minima is, where the second derivative is 0. So that's what we're showing here. We're showing the second derivative of your chromatogram. It looks beautiful. Where the second derivative is 0, that's the inflection point in your peak. Then Skyline moves those out a little bit because we don't want the middle of the peak, we want kind of the base. Okay, so now Skyline has gone through your data. It has found places that look like peaks using this minimum, this maximum. Now it groups all of the peaks together. So it actually looks across your data, and in a targeted setting where you don't have things like MS IDs from doing a DIA umpire style analysis, you don't have identifications yet. Skyline's going to go through, it's going to look for the top 20 peaks for each target in your window. The peak grouping that it's doing here, it's doing the exact same thing that we do by eye. So it's looking at the elution profile. So that's the time dimension. It's looking at the start retention time, the end retention time, and the top, the apex retention time. If all of these fragments belong together, then they should all have about the same retention time. They should all elute the same. Peaks with similar values get grouped together, and Skyline widens out those boundaries one more time. So now it's taking the boundaries for all of the peak group together. So now a little bit of building on things that Brian had just said. So now Skyline has top 20 peaks, but which one is actually the peptide? This gets into that peptide identification, the peak identification. So Skyline takes a lot of different scores. Same type of thing that we do with our eyes. We're going to be doing that in just a little bit, but it's going to take a lot of these things and for each peak group, tally up a score. The top scoring peak group is what it picks to be the peptide peak. So you might notice if you're familiar with DDA scoring and like mProfit algorithm, a lot of these are the exact same. So it's looking for things like the shape of the peak, the coelution. We just mentioned how the minimum, how the start, the end, and the apex retention time should all be the same. It's scoring that. It's scoring the intensity. More intense peaks are more likely to be the peptide, Skyline says. And then it tallies up all of these scores, and that's how it assigns the right peak group. So what does this actually look like? So in this processing, when we look in Skyline, we'll see chromatograms that look something like this. So in the Skyline software, on the horizontal axis, it's the retention time. That's the time the peptide's coming off of your column. On the vertical, it's the intensity. Intensity being kind of a little bit a proxy of abundance. And then each one of these colored lines is another fragment that was measured for that target. And each fragment is colored differently. And then the legend tells you the fragment identification, the name of the fragment. That's the Y8, Y6, Y4. And then it tells you the mass to charge of that fragment. So here I'm showing you two chromatograms in two different situations. The first situation on the left, this is a very easy call. It's very easy as a mass spectrometrist to look at the chromatogram on the left and say, this is definitely the peptide. So we're looking at things like the coelution of all the fragments are together. It makes this kind of rainbow shape. They're all stacked. The peak is very symmetrical. Compared to the chromatogram on the right, it's a little bit messier what exactly is going on. There's a second elution of this peak group, it looks like. That could be for a lot of different reasons. The chromatogram shape is a little shakier. It has a big tail on it. So when we look at these two, ideally, we want to see the chromatogram on the left as opposed to the chromatogram on the right. So Skyline's really good at detecting when a peak group is very clean, where it's very obvious. So here's one peptide from a yeast protein. If we look at this peptide across the full chromatogram, so across the entire experiment, there is really only one peak that looks like it's this peptide. It's a very strong signal. It's a very intense signal, and the fragments are all stacked very nicely. On the other hand, like Brian just showed us in real data, a lot of times a peptide doesn't look that good. So here's another chromatogram for a different peptide. You can see there's a lot of possible signals in here that could match to the peptide of interest. Only one of them, for simplicity's sake, only one of these peak groups is going to be the right peptide. So how do we know which one's right when it's such a mess? We can use things like libraries. So if we have a library MS2 spectra, whether this is a predicted one using PROSIT, or whether this is a DDA MS2 spectral library that we've acquired previously, or it's a repository spectral library that we downloaded from NIST or one of the sources that Brian gave us earlier, now we can use something called the dot product. So the dot product is just saying, based on the library spectra, how well does your measured spectra correlate? Higher is better. The perfect number you could get is a dot product of 1. The lower the dot product gets, the less likely that the peak group is correct. It doesn't match your library spectra. So this peak group at 16.4 versus the peak group at 23.9, they're both pretty intense. 16.4 is actually more intense. And the chromatogram shape looks pretty good. But if we use the dot product, the peak group at 16.4 only matches the library spectra by like 82% match. Versus on the right, the peak group at 23.9 minutes has a dot product like a match of like 99% match. So out of these two, the right peak group is the one on the right. So this is kind of what these softwares are doing when we're analyzing DIA data. It's taking all of these things together. It's taking, if you have a library spectra, it can use that library spectra, match it within your own data and say, oh, even though this data is really messy, this peak group over here looks a lot closer to the library. And this is why it's so important, if you have a library, that it matches as close as possible to your actual samples, because the fragmentation and interferences in your samples might be different, and the same instrument, because the fragmentation energy, like Brian mentioned, is a little bit specific to each instrument. Now, that doesn't always save you. Sometimes, even if you do this, it can be important to manually curate your data, because things like interference. So here's two examples of interferences in peptides where you can see Skyline picked the right peak. Over here, it picked the wrong peak, actually, on the right-hand side. But where the peak is, there's still a fragment ion that is contributing signal that's pretty noisy. So you want all of the signals to be pure signal, not noise. So even if you run these algorithms, even if you have Skyline analyze your DIA data, it can still be very important to manually look at it and convince yourself that things that are statistically significant are really, truly biologically variance and not, for example, statistically significant because there was one of these interferences in your data. So this is something that we'll be talking a lot more about later in the program as far as doing statistical analysis, but it always benefits you even after you do a statistical analysis and you get the things that are statistically significant. It's always nice to go back into the data using something like Skyline and convince yourself before you send the biologists off on a wild chase for a protein that's not actually differential. Convince yourself that the chromatograms are real. So we talked a little about these criteria for picking signal. It's the same thing we do as the expert. It's the same thing that Skyline is doing behind the scenes in all of these softwares. I'm going to just review these real quick, and then I'm going to take a couple questions. So we talked about we're looking for the coelution of all of the fragments being measured. That's making the nice stacked rainbow. Talked about the peak shape. Symmetrical is best. We talked about signal intensity. The more intense the signal, the more likely it is to be the peak, but not always. That requires no prior knowledge, no previous work. If you have prior knowledge, it makes it so much easier to pick the right peak. So if you have an MS library spectra, that gives you that dot product value. If you set up retention time standards, indexed retention time calculators, that can also speed up the process and be more sure that you're picking the right peak. Not super relevant for DIA, but if you have those stable isotope labeled standards that someone had mentioned in the chat, that helps you pick those paired endogenous peptides a lot easier, but it doesn't scale necessarily to most DIA applications. It works really great if you have targeted applications like a PRM having a heavy labeled standard. So I wanted to pause real quick before we go on and clear out some of the questions. So the MZ in Skyline, 99% of the time when you're looking at the mass to charge, it's going to be the fragment level. Skyline does include precursor level information. You can pull in MS1 scans and look at the isotope distribution for your precursors. 99% of the time in Skyline, we're going to be looking at fragments though. We got the no enzyme question. So if you're not using digestion enzyme, sorry, for if you're doing something like immunopeptidomics or venomomics, you can add a no enzyme. You could also just skip that whole step and type your targets directly into the document. So just right on the left-hand side, you can add targets simply by typing on your keyboard, whatever peptide sequence you want to look for in a targeted setting. Should isolation scheme be the same as your settings on the Orbitrap? Yes, when you're analyzing the data in Skyline, and Brendan will be doing this more tomorrow too with the tutorial. Your isolation scheme, you should tell Skyline how it's doing it. Skyline also has an option where it can read your scheme directly from your data files. So you don't necessarily have to have that setting pre-baked into Skyline to read in your data. You can have Skyline read the isolation scheme directly from your file. Can Skyline identify how many protein groups? No. So great question from Bowen. Skyline actually doesn't do any protein parsimony step like that. And it will give you options. In Skyline, you can have options where you can add a non-unique peptide, a duplicate peptide. You can add it to just one protein, or you can add it to all of the proteins. But if you want to get back at parsimony and protein groupings, you'll have to export Skyline and, for example, put that data into something like MSstats to regroup your proteins from a DIA raw file. DIA Skyline can detect peptides and all of the proteins that it came from a DIA raw file. So you're going to be doing that tomorrow with Brendan. Yeah, I think Juan's going to get this question from Hugo on acceptable value for a dot product. The short answer, the short version, it depends. Higher is better, but there's a lot of reasons why you might get a different a low dot product, a poor dot product, but it's still the right match. So I think Juan's got the rest of those, but so that's kind of like the characteristics that if we were to analyze, for example, PRM by ourselves, by eye, it's kind of doing the exact same things that these softwares are doing, and the software is just automating a lot of this. So prior information. So we can add prior information to our Skyline document using the settings, peptide settings, and there's this library tab. And you can have as many libraries in here as you want. If you want to activate that spectral library, kind of turn it on, you just check the box. We're going to be doing this in a bit using kind of like a fake repository, spectral library. Once you have a library in here, you can hit this explore button and it'll show you the spectra for all of the peptides. So a lot of times, the quality of the spectral library will make a big difference in your data analysis. So some peptides will fragment very well. So that's this bottom right-hand side. This is an example of a really nice fragmentation spectra. What makes it nice compared to the one on the left? There's a lot of fragment ions that span kind of the full MZ range. So if we look at the one on the left at the top, there's only a couple identified fragment ions in this spectra versus the one on the bottom right has many fragment ions that span kind of the full length of this peptide sequence. So this is another thing that you can do before you even go to analyze your data. If you're doing a targeted analysis of your DIA results, you might look at the peptides that you're interested in your targeted analysis, and they go in the target pane on the left-hand side, as I said earlier. And then you can go through and check the spectral library before you pull in your DIA data. And if it's a garbage looking peptide like the top left, maybe you remove that from your analysis. You don't even bother trying to reanalyze this because it's probably going to look like garbage. So you can kind of cut down before you even look at the DIA data based on your prior information that you have. Okay, so that's library spectra. We talked a little bit about that. You can see some of the papers for how to build different types of library spectra. But let's talk a little bit more about this correlation to retention time, because we got a lot of questions about that in the last section. So retention time, why should we care? Because peak picking is a lot easier when we have retention time information. So in Skyline, this is kind of an example of a target. So you could just type this peptide sequence into Skyline, and it will tell you the precursor mass to charge value, and it'll tell you a bunch of the fragment mass to charge values. Predicting MZ values is extremely easy. Predicting retention time is not so easy. So having prior knowledge of when a peptide elutes helps your software pick the right peptide peak. This is the same if you're using Skyline or if you're using, for example, Spectronaut in those Biognosis IRTs. That's why they like when you have them. The more information the program has, or the operator has, the expert has, the more likely you're going to pick the right one. So one kind of like thing I want you to recall from this, if you take nothing else away from this section, is that empirically sourced retention time is better than predicted retention time. So we're going to go into why that is, but if you take nothing else home, the take-home message is that empirical source retention time is better than predicted. So we can look at retention times in Skyline just by selecting all the targets. It'll show you the full retention time chromatogram and show you where all of the targets are. Just kind of a nice little trick. But the way that we can get retention time information, Skyline has two options built in. The first option is called SSR Calc. This approach, SSR Calc, is sequence-specific retention calculator. SSR Calc requires no prior information. It'll just take the peptide sequence and it will add all of the retention coefficients for each individual amino acid, and then add some special consideration for whatever amino acid is the N terminal. So SSR Calc does pretty well. In Skyline, this is right built in. You can see the SSR calc visualizations in the retention time window. In the retention time window, if you go to graph, regression, score to run, it'll show you this SSR calc score. It looks something like this. So now on the horizontal, we have the SSR calc score from 0 to 50 here. And then we have the measured retention time. So this is the actual retention time for these peptides on our LCMS system. So a couple things. We've got the SSR calc score. We've got this real retention time. So now each one of these points is a peptide. Notice the SSR calc score is not supposed to be an exact value of the measured retention time. It's just a score. And the correlation of that score and the correlation of the retention time is really all it's doing. So for example, this first peptide that has a very low SSR calc score also has a fairly early measured time. The higher the SSR calc score, the more likely that peptide is to elute later, have a higher retention time. So that's really all the SSR calc score can tell you. It can't tell you a very exact, precise retention time. It just kind of gives you an idea. We can also compare how well the SSR calc score works for our data, looking at some of the metrics up in the top here, for example, the R value. But really all this can tell us is if we click on a peptide in the SSR calc score window, we click on that peptide, it'll take us to that chromatogram. This one correlates very well to the predicted retention time, but maybe we want to click on some of these peptides who had an SSR calc score that didn't correlate with what we actually measured. That might be an indication that the peak is not picked correctly. So that's really all we can do with the SSR calc score. We can kind of look at the trend between the SSR calc score and the measured retention time and try to zoom in our attention on some of these peptides that did not correlate very well. So this is a very simple example, right? But as we add more targets, this gets messier and messier. So now we get a whole bunch of outliers, and we're not even really at like DIA scales yet. This is just like a somewhat large PRM experiment. But you can see already there's some extreme outliers here. There's some things that didn't have any measured retention time. Like we should go back in our data and manually curate all of these to make sure they're picked correctly. Or we use empirical retention times. Rather than just using a predicted retention time, we can use the indexed retention time, IRT. So this is a really not an easy read paper, but I found it like a very intuitive paper, not a super tough read. The Escher paper from oh my goodness, almost 10 years ago now. It's a good one. IRT, as Brian mentioned, there's like kind of like the trademark IRT, and that's just a set of peptides to use for this technique. But IRT kind of itself is just an approach that stores a calibrated, empirically measured retention time in a library. Then we can use it for future retention time prediction. So this means for future, it means it doesn't have to be on the same LC system. It doesn't have to be the same chromatography gradient. And that's really why it's so powerful. So how does that work? So let's imagine we have 3 peptides. 2 of these peptides are going to be IRTs. This can be anything. It doesn't have to be a kit of synthetic IRTs. It can be anything in your sample. So you have 2 anchor peptides, IRT peptide 1 and IRT peptide 2. Somewhere in between falls your peptide you're interested in, peptide X. So what IRT does is it says, anchor 1, set that arbitrarily to a value of 0. Anchor 2, set that arbitrarily to a value of 100. So now we have 2 anchors, 0 and 100. Whenever peptide 1 elutes, that's value 0. Whenever peptide 2 elutes, that's 100. Peptide X elutes 60% of the way between these 2. Because it's 60%, we'll assign it IRT value 60. Now, if I change my gradient, the percentage, these index values, doesn't have to change. I can change my gradient, and even though IRT peptide 1 now elutes at 10 minutes instead of 5, and IRT peptide 2 elutes at 50 minutes instead of 25 minutes, if we set those still at 100%, we know that peptide X elutes 60% of the way in between on this linear gradient, and therefore has a new measured retention time value of 34 minutes. So that's really all IRT is doing. You can set these red anchor peptides to be anything you want, as long as you measure them in both runs in all of your mass spectrometry acquisitions. As long as those anchor peptides are there, you set up the calculator one time, and then after that, you can apply that calculator in any other samples. So Skyline can set this up. A lot of the other DIA software also kind of performs a similar thing. For example, Spectronaut, I mentioned those diagnosis peptides you can purchase and spike those in to use as IRTs. Other software will just use any peptides that are measured reproducibly in all of your samples and align the retention times using that. It's pretty clever. So in Skyline, you have this IRT calculator and you set this up. When you're doing your data analysis for DIA in Skyline, this is kind of done for you. It's kind of folded into a tutorial, but this is what it looks like. And this is just taking those IRT peptides. You put your standard peptides, that's the red ones, you plug those in. You set up the calculator, assigning them a value, and I can link a tutorial for you if you're interested in setting this up yourself. And then you measure all of your peptide X's, go down in this measured peptides window. And then it just, in your calibration run, it'll assign the IRT values to your standard peptides just arbitrarily, 0 to 100. And then for your measured peptides, your experimental peptides, it'll just plug those in based on that linear fit on your lines. So it's pretty powerful. Using IRT, you see these in Skyline the exact same way we saw the SSR calc, except now instead of having SSR calc score on the x-axis, on the horizontal axis, we have the IRT value. The IRT value, like I said, can span whatever you want. It's not supposed to be a direct one-to-one value with a measured retention time. It's just to get the correlation. You might notice right off the bat, if we compare that back to the SSR calc score, look, these peptides just fall a lot nicer onto this line. And that's the exact same thing that Escher found in her paper, where the SSR calc retention time, just based on sequence prediction, is not as tight as using this empirical index retention time. So that's kind of why a lot of the DIA approaches prefer if you use kind of like this IRT style or build in some way to include retention time as prior information in analyzing the data. Okay, so we're going to switch gears here in just a bit. So let me just double check any questions that we have open. So Susanna asks, does IRT work when your retention times do not change linearly? It does get a little trickier, but Skyline does have other fits besides linear fits. So you can definitely play around with that. I think most people do not get quite as fancy with their liquid chromatography. So first, I commend you for doing nonlinear gradients and maybe more interesting multi-step gradients there. But yes, Skyline has nonlinear options for retention time calculators. Adding on to a question, what is the difference between remove duplicate peptides and remove repeated peptides? So if you remove duplicates, it'll leave a peptide in one protein, the first protein. If you remove repeated, it'll remove any peptide that exists more than once in the document from everything. So for a lot of targeted work, we like to remove those. For a lot of DIA work, you might not want to remove those because you want to do post-processing that's going to regroup your peptides into proteins. I allude to manually curating peptides. It seems reasonable in PRM when there's maybe 100 peptides, but wouldn't we let the software do this with DIA? Absolutely agree. That's exactly what I do, Leanne. I will process all of my data all the way through statistical testing, and then anything that's significant statistically, which tends to be closer to like the 100 peptide limit for manual curation. Anything that's statistically significant, I'll I'll go back and double check those chromatograms just to make sure that it's real signal. Sometimes it doesn't look great and it's not something I would want to send a grad student or a postdoc chasing for their research project until I convinced myself. So absolutely agree. Manually curate afterwards. Sometimes what I'll do with DIA is I'll spot check a couple that I know should be interesting in my data, just to make sure that the software looks like it's working correctly. Can I use a Spectronaut library in Skyline? Skyline's library building takes a whole mess of different inputs. So I will find a link or maybe one of the TAs can back me up and get you a link to the blib bibliospec blib builder list of software that it will take for spectral libraries. Question from Hugo, if I calculate the slope and the axis, intercept, yeah, on my system with IRT, can I use these values instead of IRT? You could, but I think you still need to kind of use those index values. That's, I mean, that's kind of how IRT works, is it's taking that slope and it's taking the fit of these anchor peptides, refitting those anchor peptides, and then plugging in where a experimental peptide should fall on that line. So I think you're describing exactly how it works. I think so. If you had more questions, let's follow up again later. But yeah, I think you're just describing exactly how IRT works. So good intuition.
Lindsay, I just wanted to add one thing with the library. I just shared the link, but I actually don't think this list has been updated in a while because I don't see MS Fragger there. So there might be the possibility that there might be some tools that actually Skyline supports and has been discussed in the support forum, but has not been updated to this list. So give it a try or ask in the support board.
Yeah, totally agree. The support board, hopefully Juan can drop a link somewhere. The support board, search your questions there because odds are someone has definitely asked the same question before, and they're usually very quick. Brendan and his team are very quick to get answers back to the community. Okay, so I think I'm going to move on a little bit just because a couple of these questions I think we're actually going to get to when we do some of the hands-on. Okay, so that's IRT. Skyline will take any data from any vendor. So once you get these things set up, you get your spectral library set up, you get your IRT calculator set up, you can pull that data in from any vendor's software and get your data processed. So that takes us, now your data is in Skyline, you're going to do some core analyses. There's a couple built-in native visualizations in Skyline that make it very nice for curating your data. So we already talked about looking at chromatograms. Skyline is really kind of the bread and butter of Skyline, is being able to visualize these chromatograms and manipulate them. When we look at this in the Skyline software, it looks something like this, where on your left-hand side, you get all of the targets for your experiment. So you set these up prior to bringing in your data. You have to have the target set before you can import your data, or else Skyline doesn't have any chromatograms to extract. So you'll see that it's built like a hierarchy. It's got this tree where it's the protein. Underneath the protein is all of the peptide sequence. Underneath each peptide is a precursor. So this is the MZ value for that peptide sequence with that charge. The 2 pluses here means it's a +2 charge precursor. And then underneath the precursor, you have all the fragments that you had set up in your settings, transition settings. When you click on a peptide or a precursor, it will show you the chromatogram for that target. And like I said before, the retention time on the horizontal, the intensity on the vertical, and then each one of the colors corresponds to a different fragment ion. I do have an aside here too. You can show the precursor ions as well instead of just the fragment ions. There's a whole tutorial for that that I could link if you're interested in doing that kind of thing. So we have our process data. We talked about these 3 things that we're looking for in our data, talked about how adding this prior knowledge is very important, especially for DIA, to have library and to have retention time information. We talked about how interferences can be an important reason why we need to look at our data. We might need to remove those interferences in order to get good quantification values. Skyline's really good at picking easy peaks. However, DIA does not have easy data. So how can we be sure that what Skyline has picked, or what really any of our software has picked, is real? Using some of these visualizations helps us very quickly pick out, even if we have a lot of targets, when something might have gone wrong. So right now I'm showing you a retention time replicate graph. So this means the retention time for that peak group is shown on the vertical axis. The box plot represents the start, end, and apex for that peak that was picked. Each one of the columns here is another replicate, another data file that we acquired. So you can see for the majority of these data files, the retention time is about 31 minutes, give or take maybe 30 seconds, except for one. One of these replicates is an outlier. And this particular replicate, the peptide eluted way late. If we click on that box, it'll take us to that chromatogram. And we might want to double check, did that peak get picked correctly? Is that peptide really eluting several minutes later than the rest of this cohort of samples? If not, maybe we want to look at the 31-minute retention time window and see if there's a peak there that did not, for whatever reason, score high enough to be picked. Along the same lines, we can look at the peak area percentage. So this is showing, the stacked bar chart is showing you each fragment that we measured for this peptide, and it's showing you the percent breakdown of the peak group. So for this particular peptide, the largest, most intense fragment is the red Y6, and then the teal Y4, and then maybe kind of a 3-way tie between the green, the orange, and the blue, with the purple Y9 making just the smallest percentage of the peak area, except for this one replicate. So this one replicate has a very different fragment distribution. There's almost no red Y6 in the peak group that was picked for the sample, combined with the fact that it was picked so much later in retention time from the others, that would indicate something that I would want to manually follow up on and make sure that this peak is picked correctly. This can be very important downstream when we're trying to do things like MS stats, because MS stats, we're going to be trying to compare the variance between 2 sample groups. If some of the variance in a sample group is due to a mispicked peak, it can make it impossible for us to detect that statistically significant biological variance. So this is just to show you that even if a software is doing a really good job picking peaks, going through and manually curating for some of the things you care the most about can be very important in getting the results that you hope to get. So that's just a couple of these manual curations, and I think we're doing that a lot more tomorrow too. But last, before we moved on to doing some Skyline ourselves, I wanted to point out there's a lot of additional analyses for doing things like biological inference and visualizations and statistics. So there's just a whole slew of different tools here that you can add on, kind of like apps on top of Skyline. To get these, you can add these tools just by going to this tools tab up here and going to the tools store. And if you go to add, you'll see add from tool store has a whole bunch of different options here that can be very helpful in your research. For example, I had seen one person ask a question about isoforms. This population variation tool in the middle there takes data from, I think it's dbSNP. So if you're in the clinical realm and you're looking at some particular protein and you want to make sure that you're measuring all of the peptides and single amino acid variant isoforms in the population, in the human genetics world are included, you can use that tool to very quickly do that. There's a whole bunch of other tools, including tools by Brian, including tools by Olga. So definitely something that's worth checking out. so that you could do things inside of Skyline. Okay, so that's kind of reached the end of what we wanted to mention as a very quick wrap up for this kind of lecture-based section. Targeted assays or DIA assays, Skyline handles them both. Vendor neutral, no matter what instrument you're on, you can analyze your data. A whole slew of additional software that you can add on top of Skyline-like apps. Building in tools like IRT calculators and spectral libraries. We didn't even really get a chance to talk about some of these other community-based tools, including Chorus and Panorama. Panorama for sharing your Skyline documents. But there's a whole bunch of resources on the Skyline website. So definitely check it out. Check out the support boards like Juan had said. And kind of the three main takeaways, hopefully, that you got from this is taking some of those core concepts that Brian had mentioned earlier, putting them in context in the Skyline software and what they mean in Skyline, thinking more about the quantitative LCMS data, and especially why it can be kind of challenging with DIA as far as interpreting chromatograms and processing that kind of data. And then lots and lots of literature in there. Hopefully this gave you a lot of jumping off points from your own work. And main takeaway message, let's now try to apply some of these things in Skyline. While I get that booted up, see, thanks to the TAs and Brian for getting to a whole bunch of the questions. I'm going to get Skyline booted up now. So I'm just going to open up Skyline. If you're using Skyline daily, you will probably see a whole bunch of these pop-up windows. Skyline Daily, just to clarify, is kind of like the beta version. It's like the latest, greatest, all the newest things that Skyline has incorporated into the software. New features, bug fixes get included in Skyline Daily. Some of you, it sounds like, had some issues with downloading Skyline Daily, and that's because typically you don't have administrator rights on your computer. So hopefully you found a stable release that you could do. I'm going to tell Skyline to remind me later. And for this tutorial, okay, so for this tutorial, we're just going to do kind of a pretend example. This is really to kind of get you familiar with the Skyline software, because tomorrow Brendan's going to like take you into the deep end and go straight to analyzing DIA data. So let's build a little bit of a level of comfortability of using Skyline. In this little mini tutorial, we're pretending we're doing a targeted analysis of these 6 different plasma proteins. So let's imagine, you know, your boss says, I want an assay for these 6 proteins, or let's say you have, you know, you want to go back and reanalyze some plasma DIA data. There's this, maybe an awesome paper just came out. They've got this plasma or this CSF DIA data set, and you're interested in these 6 proteins and you want to reanalyze their data for your 6 proteins. What are some of the first things that you would do to set up Skyline? So the first thing, the very, very first thing you can and should do is to go to Uniprot and get the FASTA. If you don't already have the FASTA for these, it's like kind of the basis of almost all the mass spec that the world does is get the FASTA. So you navigate to uniprot.org. If you want to play along at home, feel free to pull this up on your monitor. If you want to watch me do it and then do it again, the tutorial has screenshots and step-by-step in the PDF. So feel free to, if you want to watch one and then do one yourself later, go for it. If you're a hands-on learner, I'll try to narrate everything I'm doing. So up in the top left corner on the uniprot.org website, there's this retrieve/ID mapping. If we click on that, it takes us to this page where the very first thing it says is provide your identifiers. So here we just type in all of the proteins we're interested in. So I'll type in ApoA1, ApoA2, ApoB, ApoC3, ApoE, and KNG1. If you want, if you have your own pet protein that you'd like to do this on, feel free to type in your own pet proteins that you're interested in. Scroll down to the bottom where it says this to select options. What we have just typed in are gene names. So we'll scroll down in that drop-down menu and select gene names. We want to map this to the UniProt KB and the organism. If we start typing in human and we wait a hot minute, it should pop up Homo sapiens. So again, screenshots walking you through this are available on the course website. We'll hit submit. So you may notice it mapped to 47 UniProt KB IDs. On the left-hand side, we're going to filter by reviewed. So this is the SwissProt reviewed, and now we have our 1, 2, 3, 4, 5, 6. We'll hit download button, kind of in the kind of top leftish, middle leftish, I guess. Download button, make sure download all is selected. The format is going to be this FASTA, the canonical. You might notice if you open this up, you can do canonical and isoform. If you want to play around with that during this tutorial, feel free to do that. I'm just going to do the canonical. I'm going to download it as uncompressed because I'm lazy and I don't want to unzip it after I download it. Hit go, and you should see it downloaded. FASTA looks like this if you haven't seen a FASTA before. The first line of the sequence has the identifier for that protein, has like a short description of the name of the protein, then it has details like the organism. And then the next lines after that are the protein sequence. So it has that with this little kind of like side caret delimiter for each individual protein in here. As you're scrolling down, you might already notice some of these proteins are quite large and some of them are not so large. ApoB is just quite large, many lines of amino acid sequence. Okay, so we have our FASTA. Remember where you saved it. You might want to rename it. I think a lot of proteomicists like to rename their FASTA files, for example, by including, you got it from UniProt, Skyline tutorial. This was 6 entries and it was accessed on 2021-0503. So a lot of times, you know, later down the road when you're looking to publish, a lot of times the FASTA, you want to have the details, how many entries were in your FASTA and when you accessed it. So saving those right in the file can be very handy in the future. Okay, got our FASTA, ready to go back into Skyline. We're going to open up a blank Skyline document. So we're just going to go blank document right here. Now you should open up, we had seen this window earlier too, but in this Skyline document, what we're going to do first is go to settings. Make sure you have the default settings checked. I'm going to double check that I do. We're going to go settings, peptide settings. This is just like how we had talked before. So peptide settings, you want this to match whatever your experimental system was. You notice there's a whole lot of options here, but you can also add. So someone had a question, can you add different types of enzymes together? This is where you could kind of program that in. Maximum miscleavages, you can set this to whatever you want. For now, for this tutorial, let's just say 0. We're not going to set a background proteome for this tutorial. You will be doing stuff like this tomorrow. Retention time calculator, we don't have any. For the filter, let's change this just for fun. The default settings are 8 amino acids to 25 amino acids for the peptide sequence. Just for fun, we'll change this to 7. And we'll change the exclude N-terminal amino acids to 0. This default setting is at 25 because a lot of times, if you noticed when we were scrolling down through that FASTA file, all the proteins started with that initiator methionine. A lot of times all of that stuff kind of gets cleaved off, or it's signaling peptides at that end of the protein. So excluding those N-terminal amino acids, a lot of times biologically they're excluded anyway, so we just exclude them out of the FASTA. Go to library. We don't have a library yet. We're going to do that in a bit. The default modification is carbamidamethylcysteine. We'll leave that in there. You may notice I have just a whole slew of other modifications in here for other Skyline work I do. For the maximum variable mods, we'll go ahead and set that as 1. You can also leave that in your document if you want. We're not going to touch the quantification. We'll hit OK. And now we'll go settings, transition settings. So notice this is a lot of settings up front. Settings, transition settings, we'll go to filter. For the precursor charges, we'll do 2 and 3, pretty standard. Because we have the +3, we'll add ion charges 1 and 2. For ion types, we'll add B's and Y's. And then for product ion selection, we'll leave from MZ precursor, but we'll change the 2 to last ion -1. And then we'll hit OK. So now that we have all these settings configured in Skyline, we'll go perform an in silico digest. So those settings are giving Skyline the parameters for performing an in silico digest. So if we go to file, import, FASTA, navigate to where you have your FASTA. I just saved mine right on the desktop. I named it Uniprot Skyline tutorial 6 entries. We hit open, and you should see the target list on your left populates with a whole bunch of peptides and proteins. If you look in the bottom right, I've got 6 proteins, I've got 326 peptides, 651 precursors, and thousands of transitions. So if I were to analyze this DIA data, I could try just pulling the DIA files directly in at this point. It would be a real pain in the butt if I were to do that because I have, as was mentioned in the Q&A, we've got hundreds of precursors, hundreds of peptides that we would be trying to manually curate out of our data with no spectral library, with no retention time information. This would be a real slog. I would not want to do this. So instead, what we'll do next, let's now pretend, let's open up a new Skyline document. Let's now pretend that we have some prior information. I'm personally not going to change my save my changes, but you can if you want. Now we have blank Skyline document again. Now let's pretend we have a spectral library. We'll go to settings, peptide settings, library, go to edit list, and go to add. So we're going to pretend like we downloaded a spectral library, for example, from NIST. So we'll just call this May Institute SpecLib. This is just giving it a nickname in Skyline. And now the path, we'll go to browse. And we're going to browse to wherever you downloaded these data files, the tutorial files. So if we navigate to there, we should have, just finding where I saved mine because it wasn't on the desktop where I usually do. Okay, there we go. We have now moved these to the desktop. If we navigate to the plasma spectral library folder, I already have a blib in here for you. So navigate to the tutorial files in the plasma folder. Inside the plasma folder, plasma spectral library. If you open that up, you should have this NEU, Northeastern University Skyline plasma fraction blib. Hit open, and then hit OK in the edit library window. And then hit OK again. And then in the peptide settings library tab, check the box for the spectral library that you just added. So now you should have this set. Box is checked, library set up, hit OK. And you should see kind of this library match window pops up. I'm going to move that just in the middle so it's a little bit bigger. Oh, you may have noticed I clicked and dragged that window. If you click on the tab and hold it, you'll kind of get this blue box that opens up. And then if you're still holding it, there's tabs on either side. So there's this kind of window over here. If I click and drag and hover over that, it'll snap it to the right. If I click and drag, I can snap it to the bottom. For now, I'm just going to click and drag and set it to the center. Kind of handy thing for when you're working with Brendan tomorrow to move the windows around to match what his Skyline looks like. So we don't have any targets in here now. We have this spectral library, but we don't have any targets. So we're going to do the same thing we did before. We're going to go File, Import, FASTA. We're going to go back to wherever that FASTA was. For me, I just put it on my desktop. Uniprot, open. So notice again, now the left side target list window has populated, but it's a lot fewer things now. So if you look in the bottom right, we still have the 6 proteins, but instead of having 300 something peptides, we only have 141 peptides. If you click on a peptide and go, so if you click on a peptide, you open up the precursors by either clicking on this little plus sign, minus sign, or you can use your arrow keys. So you can use your arrow keys to tab up and down, and then use the right arrow to open it up, left arrow to close it. If you open it up, go down to a precursor, now you can see the spectra. So if you were doing a targeted analysis of DIA data, you might go down through some of these spectra and delete stuff that looks like junk. So far, all of these spectra look, not awful. So if we kept going down, tried to look for some spectra that looked really awful, maybe this guy, this particular peptide, AKP, it's not the best spectra I've seen. Maybe I would delete that so that I don't even bother pulling that data in because it looks kind of junky. So this kind of gives you an idea of how adding prior information gives us a better chance of analyzing our data quickly and correctly. So we went, if you had no prior information, just the FASTA file, and you tried to do this kind of analysis on just 6 proteins, you would have hundreds of peptides that you would have to curate and Skyline would be trying to pick from. If you add some prior information like this spectral library, now Skyline knows about half of those peptides that are theoretically possible are not empirically measured. So the more information you add, the better your DIA analysis will be. See, here's some more peptides that definitely have maybe worse looking spectra. So I would just hit the delete key and remove those without doing an analysis. So there are a couple questions. See, you guys are doing a great job grabbing questions for me. So talking about those duplicates. So we could go up here to the upper left hand, go to Refine, Advanced, and now you have options to remove repeated peptides or remove duplicate peptides. If you hover over that text, a lot of times Skyline will have help messages for you. So if you hover over remove repeated peptides, it will remove all the peptides except for the very first occurrence. So the very first protein in your target list will have that peptide, and it'll remove all the other ones. Removing the duplicate peptides, if you hover over it, any peptides that are not unique will be removed. So out of curiosity, we can just hit that, see if there's any duplicate peptides in our mini list. There were no duplicate peptides.
Lindsay, before you move on, there was a question that is relevant to the window you had open about deleting peptides without a spectra, but
yes,
by math.
So in here, there's this final option too, where you can remove peptides that do not have a library match. We can click that. The way we set up this Skyline document, every peptide has a library match. If you set up your Skyline document differently, you could click this, hit OK, and it would remove any peptides that happen to be in that target list that did not have a library spectrum. But if you go view, if you go to settings, peptide settings, and go to the library tab that we were just on, here we have pick peptides matching library. That means Skyline's only going to add peptides to this document that have a library spectrum. That said, if we did something like pick peptides matching filter, you see that the number of peptides increased by like 50. So now we have a ton more peptides, and not all of them have a library spectra. So you can tell which peptides have a library spectra or not on the left-hand side in the target pane. If you look real close, and this might be real tiny on your screen, I wonder if I can, it's already as big as it gets. So on the left-hand side, if you look real close to where there's a P and an oval, and then kind of almost looks like an antibody or something, like a little red V underneath it. If you look immediately to the next side of that, some of the peptides have a little black and white spiky boy next to it. The spiky boy indicates that that peptide does have a library spectra. And you see these first two peptides for me on my document do not have the little black spiky lines. That means they do not have a library spectra. So if I open that up and I click on these precursors, I get nothing. Versus when I go down to these precursors, I see the library spectra. Up here, nothing. Down here, no. So now if we did refine, advanced, remove peptides missing library match, those disappear and get refined out of our document. Svetlana has a question. Why did we skip miscleavages? Just for fun, we can add those back in. We can go to settings, peptide settings, digestion, max miscleavages 2. So we see nothing happened in this document. I think what we have to do is settings, peptide settings. Change back. So remember before we did settings, peptide settings, and set it to filter. If we set it back to library, there we go. So you can add as many miscleavages as you want. The more miscleavages that you add, especially when you're talking about stuff like DIA, you just increase the search space, and it can get very slow to process that data. Aaron has a question. In Skyline, once you have your target list populated and all your sample results uploaded, is it possible to filter out peptides within a protein by correlation with each other? So there are some advanced refinement things up here in Refine Advanced that you can filter out based on things like, oh, I don't have it in here because I don't have data in here right now. I don't have data in this one, but I might have another document that I can just open up to show you real quick what that might look like in another example. So here's a slightly more advanced Skyline document, but that's okay because you're going to be doing all kinds of crazy stuff soon, right? Okay, So slightly more advanced Skyline document, but it's something that I had on hand. This Skyline document, if we go refine advanced results, we have a lot more options in here to refine our results. So once you get actual data in your Skyline document, a couple more options like kind of unlock for you. So there's not really anywhere in here to do a correlation, an abundance correlation. So you would have to export this, do it in Excel, and then refine your peptide list here. But there are some other things in here that are maybe kind of nice, like refining by the CV or refining by retention time correlation. There is nothing for abundance correlation, however. So that one you would have to do outside in Excel, pull your data back in. It's not awful to do that. If you do that, you can file export, or there's an option in here, view document grid, and then you can change your report setting to peptides, for example, and it'll have the abundance measure for different peptides. You can hit this export button in the document grid to export, and then edit, or sorry, edit, insert peptides, and you can just insert whatever peptides you want to keep in there. It's definitely a workaround. So there's nothing directly for abundance. Andrew Reed, how is abundance measured in Skyline? Abundance is the MS2 chromatogram. So let's see if I can find a good one in here. Okay, so here's maybe a decent example. Let me make this bigger for you. Not a decent example. Here we go. This guy looks good, but has a giant interference. So Skyline will take the sum of the MS2 fragments and subtract the background area. So all of these chromatograms that you see in Skyline always start at 0. That's not because it's truly from your instrument starting at 0. Skyline's performing background subtraction and then showing you the background subtracted peak areas. When you're calculating these peak areas, Skyline's also including any interferences that are in here. So this big purple guy looks awful. Probably we should try to find where that purple guy is. It's a Y1, no surprise. And make sure he's not quantitative. So you can right click on the fragment and hit the quantitative tab. And then that fragment will no longer be used in calculating the peak area. So if I turn that on, you can see it adds a whole bunch of area under the curve to the peak area. Turn it off, and now it's not being used for quantification. Skyline's also using these integration boundaries. So you may notice on either side, the integration boundary here was a little bit wider than the peak itself. So you might want to refine your data and pull that integration boundary in closer. So another kind of argument for looking at your data and trying to determine if your data is real or not before you run off and try to do statistical analyses. Is abundance normalized? Abundance in Skyline is not by default normalized. However, you can set normalizations. If you right click in the peak area window, you'll see this option normalize to, and you have a whole bunch of options. If you have heavy peptides, obviously normalize to your paired heavy standards, but you could also do normalizations like equalize the medians or do a total ion current. And I am positive you guys are going to be talking a ton more about data normalization later on in the course. So things to keep in mind. I think that just about covered the questions, but I will give it another couple minutes because I think I saw a whole bunch of questions kind of going through there. I think the TAs got most of the questions in there.
So we have roughly 5 minutes left, so please don't be shy.
Yeah, I intentionally wanted to leave some time at the end. But yeah, this was short Skyline introduction for your work tomorrow with Brendan. We'll be diving right into doing a lot more of this and especially analyzing the data. So kind of focusing on looking at the data tomorrow. More questions came in. So I think we got Janan's question on deleting repeated peptides and deleting duplicate peptides. In my data, it didn't do anything because we didn't have any repeated peptides or duplicate peptides. But yeah, the repeated versus duplicate is whether you delete all non-unique peptides or if you keep the first instance of a non-unique peptide. For DIA analysis on the whole proteome, should I use as target all the proteins in my library? Yeah. So when you're doing DIA analysis on a whole proteome in Skyline, you would use the entire FASTA file, and your library would ideally have all of the proteins in it. So instead of doing file import and having just this little baby FASTA file with just 6 proteins, you would do a FASTA file with the entire human proteome or Arabidopsis proteome or whatever proteome you happen to be doing. And then when you make your spectral library or you make your DDA, your predicted library, whatever you're doing, use the exact same FASTA. Skyline does have ProSit capabilities now. So you can make a predicted spectral library directly inside of Skyline now. Use the exact same FASTA though. Can you see the MS injection time in Skyline? You cannot visualize the injection time, but I'll pull up this results file that I had on hand right here. You can see the points across the peak though, however. So down at the bottom here, next to the retention time, you see there's these red short vertical lines and then this little red number to the right, this little red 7. That's how many points were across this integration here. So if I made this integration really short, that number changes. If I make it a little wider, now I've got 7 points across the peak. Not great, not bad, but not great. IRT tutorial, let me grab a link for that. And I'll also post the IRT tutorial on the course page, the course Google Drive page. How does the program go from peptide to protein intensity? It just sums up. Peptides gets all the MS2 fragments get summed to a peptide. All of the peptide gets summed to protein. There are much fancier ways of doing this. For example, there's some options in Skyline group comparison that were really pulled from MS stats, like doing the Tukey medium polish. I think you're going to be learning more about that later too. Definitely hold on to those questions, how summing to protein level might be different. Is there a way of automatically filtering out transitions based on peak width? I don't think in the refinement that there's a way to do peak width.
I think from the reports you can. You just have to find the correct report where you have a column with the peak width, and perhaps that I have never considered that actually. It will be interesting.
Yeah. So this is we got like 1 more minute. Let's see if I can real quick. So if you did that view document grid, you could potentially use some of these values in the document grid. There's average retention time, but there's a whole bunch of other values that you can play with in here. So Juan is kind of getting at the idea that you could probably use some of these metrics in the report to refine using this kind of data. It's not a single button though, unfortunately. Yes, Sean, the points across the peak are the number of MS reads across that peak. Yes, from the raw data, not the resampled data. Yeah, I think that's pretty much all the time we have. But if there were more questions, definitely put them in the Google document so we can try to get there. And the IRT tutorial will be linked into that Google document. So hit refresh and hopefully that will get all of your questions, and hopefully that prepared you for the theoretical and the practical side of DIA for your session